Dear student, I am Anil Srivastava from Government BYT Autonomous College Durg. Today we are going to discuss about the application of cell culture. The topic is relevant for the fourth semester biotechnology students. Today the cell culture technology is widely accepted due to its greater applicability. Since its inception, the animal cell culture in the 20th century is related to its usefulness in research. Early methods such as the Harrison culture of the inverted drop, Carroll's and later works innovations made with the introduction of the use of the infusion pump, the culture of HeLa cells, and vaccine design using animal cell culture are approach which revolutionized the implementation and study of the cell culture. There have been advances in the cell culture model for research and drug studies. The cytotoxicity and permeability of drug molecule and delivery systems are evaluated by cell culture model both in the pharmaceutical industry and in the academia. Cell models serve as an important platform to investigate cytotoxicity and permeability study by reducing the use of animal model. Since 3D cell model mimic in vivo cell better, it plays a significant role in the testing of drugs. Studies conducted during the initial development of drugs such as toxicity, cohesion and drug activity were carried out on animal. However, in the past 10 to 20 years, alternatives have been sought due to the fact that animals do not effectively model human in vivo conditions and unexpected response are observed in the studies. So, cell culture studies made positive contribution to the initial development of drugs, contrary to animal studies. The need for low drug and a short response time are the characteristics for in vitro cell culture method. In 2005, more than 100 million animals were used and $10 billion were spent for animal toxicity experiments. It is possible to reduce the cost and the amount of animal use for experiments with well-designed cell culture studies. So, among other health authorities, the FDA, Food and Drug Association, has recommended the use of human cell line to identify metabolic pathways for drug and share their applicability in vitro test in guidelines published in 2004. In November 2013, the National Institute of Health of USA reported that more than 230 embryonic stem cell lines are appropriate for this research. So with the advancement of the biochemistry, molecular biology and related subjects, now it becomes very much feasible to introduce cell culture method to resolve the problem of many health side sector, agriculture side sector and some tissue engineering sectors also. Here, what are the basic cell culture research and why the basic cell culture research are being used in which fields? This is the summarized data that the basic cell culture research are applicable in the field of the DNA transcription, protein synthesis leveled with the radioactive isotopes of fluorescence study, metabolism study using immobilization techniques, a specific cell lines study, issue of cell cycle and senescence, characterization, proliferations, and differentiation and apoptosis studies. The application of cell culture is very much useful. Besides these, there are some others also, like the studies of the biomolecule intracellular flow as RNA processing, protein transport, assembly and disassembly of the microtubules, modeling cell lines for research, etc. In the field of genomics and proteomics, this technique is very much useful in the field of the genetic analysis assay, infection, cellular transformation, immortalization, gene expression, metabolic pathways, cellular interaction as morphogenesis, proliferation and additions. There is a very big historical background related to the cell culture technology, But first of all, we can't forget the William Rocks in 1887. First of all, he tried to maintain the living cells of the neural plate of chick embryo in slime buffer for a few days. Thereafter, a long history in 1907 by the Ross, Granville Harrisons, in 1910 by the Montrose Burroughs, in again 1910 by the Montrose Burroughs for another cells, the chicken, rat, and dog and human tumor cells. The most important development in this field came in 1951 when Dr. Jones diagnosed Herenita lab with cervical cancer using a cervical biopsy. This new cell line called HeLa cells gave him the best means to develop the poliovirus, allowing the development of the SARS vaccine also. Sage Henrietta Lacks in this photograph is George Otto Gray, the surgeon of the Henrietta, transferred the cancer cell, cervical cancer cell, to the Otto Gray, and finally he maintained the cell lines. And even today we are using the HeLa cell line for a lot of experimentations, and the HeLa cell line have yielded so many findings in the field of the pharmaceuticals and others. What is its basic applicability? In a nutshell, this diagram of your book nicely represents the applicability of the tissue cell culture, like the intracellular activity, intracellular flux, pharmacology, cell-cell interaction, toxicology, tissue engineering, genomics, immunological field, and the cell products like the proteomics, secretion, and others. So this diagram beautifully explains the diversity of the cell tissue culture applications. And besides this, the production of the antiviral vaccine, understanding of neoplasia, transfer of DNA to culture cells, monoclonal antibody productions of human growth hormone like the insulin and interferon, etc. Stem cell culture differentiation into neuron, implanting normal fetal neurons into patients with the Parkinson's disease, homografting and reconstructive surgery using individuals' own cell by the tissue engineering method, and in vitro fertilization. These are the major output of the cell culture application. But three fields are very remarkable. These are the viral vaccine development, monoclonal antibodies development, and recombinant protein development. For the viral vaccine, from the ability to grow virus in the cell culture, and monoclonal antibody from the ability to generate hybridoma by the cell fusion. You all know hybridoma technology. Regarding recombinant protein, from the ability to transfer cells with isolates gene and amplify to allow high level of expression of the corresponding proteins. These three are major achievements of the cell line applications. First, the viral vaccine. In 1949, Enders discovered the poliomyelitis virus may be developed in the monkey cell line, and the polio vaccine produced in 1954 was the first human vaccine to be produced using large-scale cell culture techniques. This eventually led to the development of animal cells technology for the production of a range of human and viral vaccine against a variety of disease. Later, scientists have developed so many cell lines for the propagation of virus in chick embryo, and chick embryo is being used widely for the development of this vaccine, such type of vaccine. Here are some major achievements in the field of the vaccine development by using cell lines for the human and veterinary are as follows. Like for the human, already we have developed the vaccine against the polio, measles, mumps, rubella, yellow fever, rabies, and influenza. For veterinary, against the foot and mouth disease, Parker's disease, Newcastle's disease, Rinderfest rabies, and canine distempers, swine fever, and gluten, fowl pox, etc. India is one of the leader in the vaccine production in the world, and there are three major vaccine types have been established till date. One is an inactivated pathogenic virus, a disease causing virus which has been chemically inactivated by this method. The second method is an attenuated live virus. In this case, this is capable of propagation, virus is capable of propagation, but has been changed genetically to that it cannot produce a disease. And third is by using the recombinant DNA technology. Many of the vaccines are being produced recently. All three methods, the attenuated method and RNA-based technology and DNA recombinant technology, all three methods are being used for the development of coronavirus across the. Here, regarding cell line, safety is one of the major concerns. In 1954, the regulatory bodies in the USA decreed that human vaccines should be produced from primary cell derived from the monkey for the development of polymyelitis vaccine. However, it was discovered later that these cells were contaminated with nearly discovered tumorigenic virus designated as SV40. And this caused concern even though there was no evidence for the transmission of SV40 virus to recipient of the vaccine. After extensive study, the normal diploid fibroblast of the human was used for the development of polio vaccine, and for that in 1960 two types of cell line were developed, WY38 and MRG5 cell lines. The Vero, the African green monkey cell, was the first continuous cell line to be accepted as substrate for human vaccine productions, and polio vaccines has been produced from this cell line in France since 1983. This is a diagram. By this diagram, this is from your book. By this diagram, you can easily understand that how by the help of the this technology, the HBAS gene may be expressed in the yeast. Just HBAS gene through the help of the EcoRI transfer to the vector yeast, and one competent yeast cell was developed, and then it was used for the production of the vaccine. In this second diagram, this is also from your book. A vaccine was developed for the foot and mouth disease in the cattle. Here the RNA core was used for the synthesis of the complementary DNA, and by the help of the plasmid, one R plasmid was developed, and in the E. coli one transformed E. coli was produced for the production of this vaccine. This is in practice already. This is one new. Here the mosquito cell culture to detect the dengue virus. The fluorescing cells seen here are infected with the dengue virus. So this cell culture is here applied for the development of vaccine against the dengue fever. One is the monoclonal antibody. Here, all useful application of monoclonal antibody depends upon their high specificity of binding to an antigen. MABs also have applications as therapeutic agent, particularly in target drug therapy. In 1986, the monoclonal antibody named OKT3, developed by one pharmaceutical company, which recognizes the surface antigen CD3 on T lymphocytes and is one of the most effective agent in preventing immunological rejection of transplanted kidney. Various monoclonal antibodies have been targeted to membrane-bound protein antigen specifically expressed in tumor cells. These antibodies can be designated in configuration likely to cause the destruction of target tumor cell. The conjugation of radioactive and toxic compound to the antibody can result in a localized high concentration, resulting in cytotoxicity to the target cells. Alternatively, an enzyme may be conjugated that will catalyze the release of toxic product from an ingested prodrug an effector function through the use of a biospecific antibody that could potentially activate T cell, leading to the specific destruction of target cell. But in the case, unexpected toxicity was reported due to human anti-murine antibodies immune response. So the development of human chimeric antibody in the 1990s increased rapidly the rate of licensing of monoclonal antibody as therapeutic agents. It also do not recite the human anti-murine antibody effect, and half-life was also very longer. So most mouse IgG have half-life less than 20 hours, whereas an antibody with human type constant region can have a half-life of up to 21 days. The success of this chimeric antibody can be illustrated by Retux, which is used in the treatment of non-Hodgkin lymphoma, and this has a murine variable region which binds specifically to the CD20 on B cell and human epidomain to trigger effector mechanism. CD20 is a protein expressed by over 90% of the lymphoma cells, and these tumor cells can become coated by the anti-CD20. This results in activation of the complementary pathway and FA receptor bearing cell, which can destroy the tumor cells. Here, this is a diagram. In this diagram, just you can recall that how the hybridoma, using the hybridoma technology, we can take a product. This is the next part of the hybridoma technology. Now the recombinant technology, recombinant protein development. Here, the protein extracted from the biological sources have been important for substitution therapy since 1920s. So after discovery of the cell line technology for production of natural physiologically significant protein, a specific gene is to be isolated and expressed through cultured cell for production of desired product in large quantity and purity. There are two major products by this technology, by using this technology. One is the insulin produced by the beta cell of the islet of Langerhans, and the nature of the insulin is hypoglycemic, meaning to reduce the sugar level from higher level to within the range, meaning during fasting condition in the human body it should be from 70 to 110 milligram per year, and then after taking meal it should not cross 140 milligram per year, and this range of the sugar in the body, human blood, being maintained by this insulin. And this is very serious problem that in large number of the human populations, either the defect in insulin or being produced or insulin productions are badly hampered. So first of all, it was extracted from the animal, the cow, thereafter from the pig, and nowadays it is being used by the DNA recombinant because it is being produced from the Escherichia coli cells. So by using the cell culture technology, nowadays we are producing insulin, and now we are not dependent upon on any other animal for the demand of to fulfill the demand of the insulin. The second important thing is the interferon, production of the interferon. Here the interferon are now recognized as a family of cytokines having antiviral, antiproliferative and immunoregulatory effect. The viral infection induces the synthesis of interferon by depression of the gene, and the secreted interferon can then induce an antiviral state in neighboring cells. Here you can understand that an interferon refers to an inducible secretory protein produced by eukaryotic cells in response to virus and other stimuli. So the molecular mechanism of interferon protection is not fully understood till date, but it is supposed that it may involve synthesis of number of enzymes, including those capable of degrading the viral RNA and inhibiting protein synthesis. The interferon are a group of relatively small protein that contain 140 to 170 amino acids only, and are divided into three main types: the interferon alpha, beta, and the gamma. And further, the interferon alpha is subdivided into more than 25 types. And each interferon type is derived and secreted predominantly by one cell type, that is the B lymphocyte secretes the IFN alpha, fibroblast cell secrete INF beta, and T secretes INF gamma. So its secretion is very specific for their respective cells. And the human interferon beta is specifically secreted by fibroblast in response to viral infection or to double-stranded RNA. In vitro studies have shown the ability of human interferon beta, HULFN beta, to induce or suppress over 20 gene products. Its beneficial effect are mainly depends upon the regulation of the T cell function, modulation of production of cytokine causing anti-inflammatory effect, regulation of T cells migration and infiltrations. In 1993, the use of recombinant HULFN, meaning the human interferon beta gene, beta, was approved in the treatment of multiple sclerosis. The isolation of HUFN beta gene allowed the construction of genetically engineered E. coli and CHO cell line for its productions. Here, by this diagram, you can see just how the interferon being produced after entry of the virus. This is simple expression for the production of the interferon. In one cell, the virus when entered, it starts synthesis for a specific protein which comes and binds with the another neighboring cells for protection of the cells from the same virus. The next major product of the cell culture is the plasminogen activators. Today, thrombosis is a major problem, not only in the Western country but also in our Indian subcontinents. Sometimes internal clotting takes place inside the blood vessels, which is against the natural process, and there are so many factors have been identified to induce the internal thrombosis in the circulating blood vessels. It causes several kinds of the pathogenesis like the heart attack and the brain stroke, etc. So to solve this problem, again our cell culture is very important, plays important role. One factor, the tissue plasminogen activator TPA, is required for conversion of plasminogen into the plasmin, and the plasmin in our blood, soluble fibrin are floating. So plasmin act on the soluble fibrin, convert it into insoluble fibrin, and thereafter coagulation process takes place. So by the help of the cell culture, we can produce this plasminogen activator. The large scale production process of tissue plasminogen activator was originally developed by using the CHO-KI cell line. In 1987, TPA was one of the first recombinant products derived from mammalian cell culture and approved for the human therapy. Next one, the blood clotting factor. Although more than 30 types of the blood clotting factors have been recognized, but factor 8 and factor 9 are very important, and cell culture technology is very competent enough to produce the factor 8 and the factor 9. Regarding factor 8, the transfection of mammalian kidney cell line, the BHK, with an expression vector containing the constructed gene allowed synthesis of the biologically active molecule with the correct tertiary folding and glycosylations. Here the recombinant factor 8 secreted by these cells can be stabilized by the addition of von Willebrand factors. The two factors are normally found as a combined protein complex in blood plasma and are both essential for blood clotting in vivo. The large-scale process of factor 8 production, presently operated by some other industry by using the BHK cell line, containing approximately 150 copies of the factor 8 gene per cells. Regarding factor 9, the gene for factor 9 was originally cloned in 1982 because the protein requires glycosylation and gamma carboxylation for full activity, and for that the rat hepatoma cell is being used. The next is the erythropoietin. You may consider the erythropoietin as a hormone. It comes from the kidney and acts on the bone marrow for the erythropoiesis. Erythropoiesis is a mechanism for the production of the production of the blood. Across the world, the depression in the hemopoiesis is a common phenomenon, and so many people are suffering from the poor production of the blood. So again, the erythropoietin plays important role, and it becomes essential to identify the phenomena for the production of extra erythropoietin, because the kidney fail to produce a sufficient amount of erythropoietin. That's why the person is suffering from the depressed erythropoiesis. And again, for this, the CHO cell line played an important role. Here the kidney is the source of synthesis of glycoprotein hormone erythropoietin, and the CHO cells transfected with an expression vector containing the EPO gene have been produced by Amazon USA. The next one is the tissue engineering. This is another important achievement of the cell culture applications. Here the ability to reconstitute human tissue from combinations of cell type growth in culture is an important prospect for future therapeutic treatment associated with the organ failure. Up to now, the most successful application of tissue engineering is in the reconstitution of skin following several parts. And skin is having the double mode of origin. The upper layer is the epidermis and lower is the dermis. Epidermis is made up by the epidermal cells, ectodermal cells, and the dermis is made up by the mesodermal cells. So problem is double mode of origin. Here also, by the application of this tissue culture engineering, the problem has been solved. The artificial skin now can be formed from two layers of the humans. The dermal equivalent may be derived from by using the fibroblast cells, and the epidermal equivalent may be used developed by using the dermal surface cells. Here, to construct a dermal equivalent fibroblast are dissociated from the tissue biopsy and mixed with the predetermined proportions of the collagen in culture media. The mixture is poured into a mold, the shape of which determines the geometry of the constructed tissue. The fibroblast causes the condensation of the collagen into fibrils to form a gel-like matrix, which extrudes most of the surrounding fluid. And during a 6-day culture period, the cell attaches to the collagen, forming a contracted matrix of the tissue-like consistency. So in this way, one dermal equivalent may be developed. For epidermal equivalent, it may be developed from the keratinocytes, then layered on the surface of the dermal equivalent. The dermal cells attach to the collagen substratum to form a continuous sheet in which cells can differentiate and forms multilayers. After 10 days, the epidermal cells may be exposed to air to allow the formation of an outer protective layer, which consists of a layer of dead cells. The bilayer of cell increases in thickness over time and results in a controlled deposition of matrix protein. The living bilayer skin substitute is created to heal wound and has been used successfully for grafting to burn patients. So in this way, artificial skin is now possible by the application of the cell culture, by using the application of the cell culture. But artificial organ development is still problematic. It is not very successful, but some milestones achievements have been reported, especially for the beta cell of the pancreas. And multiple cell types requires for more complex scaffold and an extracellular matrix to support the functional relationship between the cells. A further level of complexity is that multiple cell layer requires a nutrient supply equivalent to blood capillaries in vivo, and this becomes very difficult in tissue culture method to apply to substitute the blood vessel supply, blood capillary supply. However, the use of cultured cells to replace damaged cell in vivo offers some intermediate and promising possibilities. And this concept has been developed for pancreatic cells capable of secreting insulin to reduce damaged islet of Langerhans to treat the hyperglycemic patients. But another problem in this case is the immunogenic rejection. And a possible solution is to encapsulate the cell in a matrix such as alginate or agarose. And this should protect the cells from immunological rejection but still allow the free flow of nutrients and small secreted cell product, that is the insulin. So we have technology to substitute the beta cells. We have also technology for the implantation of beta cells after immunogenic protections. But for other organ development, still we are far behind. Now regarding drug screening and toxicity test. Today for the drug development, most of the in vivo experimentation has been replaced by in vitro experimentations. Although there is problem, the in vitro experiment can't give result released for the correlative physiological experimentations of different organs. In our body, if one organ is being affected, it may alter the function of the another organ, meaning the altered function of the heart may adversely affect the altered function of the kidney or the intestine or the lungs also. So the correlationship study is difficult in vitro study. It is for that we have to go for in vivo study only. But most of the experimentations related to the drug developments are now feasible by using the cell line only, meaning by the help of the in vitro study. So nowadays the first step of the drug development depends upon the in vitro study. And for the drug development, normal fields where the in vitro testings are going on are for the reduced growth rate study, breakdown of membrane permeability study, tissue specificity of response study, ability to metabolize toxic compound, stimulate wound healing process, damage repair by use of artificial constructed tissue, and genetic effects, mutagenicity, and interaction with the DNA. So these are the major parameters to prove that a particular lead compound is suitable for development of the drugs. So it has reduced a large number of the use of the animal, and now it gives very promising result also for the development of different and toxicity study. For the cell therapy, there is high turnover of hematopoietic cells in vivo as a result of continuous growth and replacement. And this makes the cell vulnerable to destruction by any cytotoxic drug used in the chemotherapy to eradicate residual tumor cells and other. So removal of bone marrow cells prior to chemotherapy allow isolation and expansion of the pluripotent stem cells to provide a source of mature hematopoietic cells following chemotherapy. The ex vivo cells explain the culture control of stem cells to allow the production of cell populations enriched with a specific cell line. And this procedure can be useful for the production of a specific T lymphocytes or natural killer cells that may be required for cell-mediated immunotherapy. So this is again useful here. And this picture just shows that how we can use the cells for different cell therapy, like the for the replacement of the brain cells, bone marrow cells, and skeletal muscle cells, and skin. But there are so many limitations also. It's not so easy. Now next one is the gene therapy. Here the development of method for transfecting exogenous gene into mammal cells has been extremely important for producing cells capable of expressing large quantities of therapeutic proteins in the culture. The concept, uh, the concept of gene therapy is that a missing faulty gene is replaced by a normal working gene, and the process involves the transfection of a specific gene into cell for a patient with an identified and well-characterized genetic disease. The gene can be introduced into cell inside the patients by in vivo methods or outside the patients by ex vivo method. For the ex vivo process, the cells that are removed are then reintroduced into the patients. Then uh, they express the normal protein which elevate the metabolic defects. However, the transfected cells themselves may also be used directly as a treatment for certain genetic disease associated with the inborn error of the metabolism. The therapeutic procedure is suitable for treatment of disorder associated with the hemoglobin, such as sickle cell anemia, thalassemia. Like other potential disease, a target for the gene therapy includes so many diseases like the X-linked recessive deficiency of the hypoxanthine guanine, phosphoribosyl transferase enzymes, and phenylketonuria, and some others also. Just this figure indicates that a virus having three genes, the A, B, C, and here A modified virus a beneficial gene, how we can transfer to other after detections. And for that, so many vectors are required. You all know that what are the vectors? These are the vectors for the transfer of the gene. So gene therapy is also very suitable and it's very promising. And another important is now important development is now the CRISPR-Cas9 technology. And I am very much hopeful by the CRISPR-Cas9 technology that it will solve so many problems. So thank you. I hope that the account for the cell culture application will be suitable for your exams and preparations. Thank you very much.